Welcome! Today, we turn our attention to a molecule that plays a stealthy yet powerful role in cancer progression. LYPD3, also known as C4-4A. This protein acts like a molecular hook on the surface of cancer cells, helping them detach, migrate, and establish new tumors in distant organs. We'll explore its expression distribution, structure, signaling pathways, its dual roles in normal healing and malignant spread, and why it's becoming a compelling target for new therapies. LYPD3 is a protein that resides on the cell surface, tethered by a glycolipid anchor. In healthy tissues like skin and lung, it facilitates necessary cell movement during repair. However, many cancers hijack this machinery. They dramatically increase LYPD3 expression, exploiting its pro-migratory functions to fuel invasion and metastasis, while often remaining undetectable in adjacent normal tissue, making it an attractive therapeutic bullseye. LYPD3's expression pattern tells a story of context. Under normal conditions, it's found in specific layers of the skin, bladder lining, and placenta. But in cancer, it's frequently overexpressed. It's prevalent in aggressive bladder and breast cancers, and its levels often climb as the tumor becomes more invasive and metastatic. This tumor-restricted overexpression is a key reason researchers are interested in targeting it. Structurally, LYPD3 is a compact, three-domain protein linked directly to the outer cell membrane by a GPI anchor. The first domain is crucial for binding its partner, the urokinase plasminogen activator. The middle domain interacts with integrins, key adhesion molecules. The third domain not only anchors the protein but can also be snipped by enzymes called MMPs, releasing a soluble version that may have different functions. This multi-domain design allows it to engage with multiple signaling systems. LYPD3 doesn't act alone. It's a central hub in a pro-cancer signaling network. By binding the UPA enzyme, it kickstarts a cascade that dissolves the surrounding tissue, clearing a path for invasion. Through partnerships with integrins, it manipulates the cell's internal skeleton to propel movement. Most critically, it activates the powerful PI3K-OCT survival pathway, which blocks cell death and promotes traits needed for metastasis. In some cancers, its own production is turbocharged by transcription factors, creating a vicious cycle of aggression. The clinical impact of LYPD3 is clear. Several hard-to-treat cancers. In kidney cancer, its high levels are linked to worse outcomes. In aggressive triple-negative breast cancer, it's a direct target of a transcription factor that promotes tumor growth. In stomach cancer, it helps cells survive the stressful journey through the bloodstream. Its consistent presence in advanced and metastatic bladder cancer further underscores its role as a master regulator of cancer spread. Targeting the LYPD3-UPA axis is an active area of translational research. Strategies include direct inhibition with antibodies like lupartumab. An innovative approach uses a fluorescent dye that binds to UPA, helping surgeons see tumor margins in real time during operations. Interestingly, because this pathway is also involved in tissue remodeling. In kidney disease, a UPA-targeting antibody is in trials for diabetic nephropathy, showing the pathway's relevance beyond oncology. More target sharing. See you next time.